extra credit. I'll give you guys until Thursday to do that, and you can just email me, that would be fine. Also, I'll be passing back the exams. I forgot to bring them with me, but I'll go grab them at a break and pass those back for the second half of class today. And so the mean on that was 74.4 again, so that's really right on target, so good job on that. All right, so for the last lecture, we're still talking about plants, and we'll talk a little bit more about patterning in plants, especially of flowers. And this again gives me a chance to sort of do a little bit of compare and contrast to what we learned about patterning in animals, so that we can see some of the mechanisms behind this. So one of our things about plants is that they do undergo morphogenesis during their entire life. So unlike animals that just have this embryo stage where they're building all their organs during that limited time in life, plants are building their organs, like leaves or shoots or roots or flowers, throughout their entire life. And a really critical structure for this is the meristem. And there'll be one major meristem right at the tip of plants known as the shoot apical meristem. And this pretty much persists throughout the entire life of the plant and is always the source of stem cells to make new leaves, new shoots, and new flowers. But in addition to the shoot apical meristem, there are some other meristems that come as well. And these are called auxiliary meristems. And you'll find these near the sides of the shoot apical meristem, but also pretty much anywhere a leaf or another shoot joins onto a plant. Right there in the little elbow corner, or right in the crux of that joining structure, is another little meristem that is, again, a source of stem cells that could easily grow into another shoot, leaf, or whatever structure. Then on the root side of things, there's also a root meristem that likewise gives the primary taproot growing down and then auxiliary root meristems growing out to the side. So these are the source of the cells that will become future organs within the plant. And so these meristems have to have some kind of information about where they are in the process and what they should do next. So there are a number of different pathways here that help stage the meristem depending on where that plant is in its life cycle. So initially there's what's called the juvenile vegetative growth meristem, and then this will convert to an adult vegetative growth. So just in the transition from being a very young, immature plant to being an adult plant capable of making all the adult structures. There are some genes that change the character of that meristem called the phase change genes. And once we get into this adult vegetative stage, now this meristem is capable of making shoots and leaves. So these are your kind of non-flower structures that an adult plant would have. Then, to get to the point where this meristem could actually make a flower, it requires two steps. First, there's a step to create a meristem capable of making a flower, and this is called the inflorescence growth. So the term inflorescence just refers to the entire flower structure, which in many plants is actually multiple different flowers. So just this entire flower structure or cluster of flowers. Or it could also mean the reproductive structures of the plant. So what we're doing is now converting this meristem from one that could only make vegetative structures to now one that's capable of making a flower or flowers. And that also takes some genes. And these are called the floral meristem identity genes. And we'll talk about a few of these: leafy, apella 1, and cauliflower. And up here in blue, you see these are different environmental signals that the plant might sense and incorporate into the expression of the floral meristem identity genes. So plants, of course, they can't move around and they need to sense and interact with their environment. And so this is the place where the environment can impact the expression of genes that tell this plant, okay, it's time to get busy and start flowering. So things that are often seen as cues to plants would include photoperiod, So many plants have to have a certain length of day or length of night to actually trigger whether or not they're going to flower. So this helps them sense what season it may be. Autonomous pathway, this just means cell autonomous mechanisms, right? Any kind of gene that's inside the cell that is not really necessarily responding to the environment, just some sort of internal mechanism that the plant has to decide its own timing in its life history. A vernalization pathway, this is a pathway where a period of chilling is required. So some plants actually need to get into a cold period before they would be able to proceed into the next reproductive stage or making a flower. And then gibberellin is a hormone that plants have, and some plants need to have a surge of this hormone in order to trigger the expression of the floral meristem identity genes. And again, push that pathway, push that meristem into a flower-capable stage. So this offers a number of ways that the plant can interact with its environment and respond to it. Then, once we get this meristem that's capable of making a flower, the next step is we're going to divide that meristem up into territories and decide which cells will make what structure of the flower. So the flower, I'll show you here in a minute, has sepals, petals, stamens, and carpels. So at least 4 distinct types of structures that are part of this flower. And there'll be some different territories within this meristem that are dedicated to each type of flower structure. And so again, there's a set of genes for this that are going to say, okay, these guys should make the sepals, these guys should make the petals, these cells should make the stamens. Okay, so this slide, I'm sure you're all pretty familiar with already, but this just gives the terms for the adult structures in the flower. The sepals are these small structures at the base of the flower, and depending on the species, the sepals may actually look a lot like petals. So they may be colored, and they may have a kind of that soft petal-like structure, or they may be just very abbreviated, small bract-like short leaves at the base of the flower. Then petals, in the simplest flower, they just have one circle or whorl of petals. And then we have the male structures, which are the stamens. So they'll be on those long structures known as filaments and anthers. That's where the microsporangia are located inside the anthers. And then in the middle, we have the carpels, which are also called pistils, and there's probably a technical difference here, but we're not going to get into it. So for our class, it's the same thing. And those are composed of stigma, style, and ovary. And then within the ovary, there may be one or more ovules where there'll be an embryo sac with an egg that could be fertilized. So plants are organized in whorls, and Arabidopsis has become a major plant model system for trying to understand the genes that are necessary to build the character and structure of these whorls. So this is the Arabidopsis flower, and you can see here these green sepals, 4 white petals. So it's not a particularly beautiful flower, but it's a very simple flower, just composed of a single ring of petals, and then a single ring of stamens, and right there in the middle, the carpel, and you can see at the tip of the carpel the stigma, which is that sticky type of structures that the pollen will come and land onto and stick to. So this is considered to be your prototypical angiosperm flower. So, and here are the basic organization of each flower. So you can see outside layer of sepals, then inside of that a whorl, a spring whorl. Back up for a second. WHORLS, whorl is just a ring, but I'll be using that term to describe these rings of flower structures. And so Arabidopsis happens to have 6 stamens, and in the middle it has kind of a fused double carpel together. And so that's one individual flower, and then the Arabidopsis inflorescens has these multiple clusters of flowers that are all coming out from the apical meristem or its auxiliary meristems. So it has a simple pattern to this structure, and this means that we could, say, do a forward genetic screen to try to find mutants where this pattern has been disrupted, and then based on that, identify genes that are critical for this whole process of partitioning out cells into the different flower structures, or even for getting the flower to the point where, or getting those meristem to the point where it could make a flower. So some of the meristem identity genes. So now we're going to look at this first group of genes, which is these here, the floral meristem identity genes necessary for this transition. And I'll show you what some of these mutant phenotypes look like. So up here in A, this is the wild type flower with its cluster of those simple flowers. And then B is a mutant in one of the meristem identity genes, floral meristem identity genes called leafy. And what you see here is these structures, these meristems, have been partly converted back into shoots. So shoots are stems plus energy or auxiliary bud structures. So basically non-flower portions of the plant. And they still have a little bit of floral character, but then they're still kind of also a little bit more like stems or shoots as well. So it's been possibly a partial conversion of that meristem to a flower making ability, but not the full extent that you would normally see. Here's another example with the Aq1 stands for Aug5Part1 mutant, and again, they can make something that sort of looks like flowers, but it also looks a little bit more like stems and shoots. So they've been compromised in the ability of that meristem to convert to a flower-making machine. And then it's also kind of interesting to see the double mutant here. So now all of the genes for leafy and Aug5Part1 have been mutated, and now this time we really see very little flower character from this meristem. It's much more like just leaves and shoots or vegetative types of structures. So leafy and Aug5Part1 may have a little bit of overlapping function here to try to promote the conversion of that meristem into an inflorescence meristem. Then there's this one, which is also interesting. So there's another gene here called cauliflower, and cauliflower on its own actually doesn't make much of a phenotype. You still see more or less wild type looking flowers. But when you combine a cauliflower mutation with Aug5Part1, and all of these are floral meristem identity genes, now this double mutant gives this phenotype here where the meristem is converted into quite a vegetative state, and all you get is hundreds of tiny little shoots. And they never make any actual flowers, but they just stay in this vegetative state of hundreds of shoots. Okay? Gives this kind of cauliflower appearance. And, so this is an angiosperm, it's a flowering plant, and if you think of some of the other horticultural crops we have, or food crops that we have, and the appearance of them, like cauliflower and broccoli, I would imagine that there are similar sorts of genes in those plants, and that they may actually be mutant, and that's why we're getting some of the phenotype that we do from those particular structures. So, back in the day, we had talked about the idea of a homeotic transformation, where you take one body structure, and instead of disrupting or breaking this body structure, you convert it into something new and different. So that one body structure is replaced, and this was homeotic transformation, and that any gene that could do that would be considered a homeotic gene. And we talked about many homeobox or Hox genes that could mutate to produce this special phenotype. So many Hox genes can do this, but not necessarily all of them. So now we're going to have a look at plants and see some examples of homeotic transformation in plants and think about what the genes are that could be responsible for that. So the first example here is, and actually the leafy newt is considered to be a homeotic transformation because instead of making flowers, this flowers really were converted into some other body part that happened to be more vegetative in nature. So they were making shoots instead of making flowers. So this leafy newt is actually considered to be homeotic. And here's another example. This time, an example of a floral organ identity gene. So now there's a second group of genes that converts the inflorescence meristem into your floral meristem. One of these genes is known as Aug5Part1. And in a wild type flower, we see a ring of sepals, a ring of petals, a ring of stamens, and a ring of carpels, or acarpal. And in this Aug5Part1, we see now these sepals have been converted into carpels. They don't look like very perfect carpels, but you can see a little bit of this pistil, or this stigma structure here. So they make these sticky, hairy cells on the tip that are characteristic where the pollen is supposed to land. And if you tested this with markers, such as genes that are expressed in carpels, you could identify these outer structures as actually being carpal in nature. Then they actually have 2 rings of stamens instead of just one. And then they have a carpal structure in the middle. So at least the 2 outer whorls in this plant have been changed from being sepals and petals into being carpels and a second layer of stamen. So that's interesting for a couple reasons. One, it's interesting because we changed one body part into another, which is homeotic. And then a second reason is, why another carpel and sepal? Why those structures and not something else? And there's a nice model that can perfectly explain that we'll talk about later. So this is also a homeotic transformation. And these homeotic transformations in plants are actually usually not due to homeobox genes. So plants do have homeobox genes, but for the most part, they're not really involved in these 2 phases of making the inflorescence meristem or dividing that up into the floral meristem with the territories. Instead, there's a different group of genes called the Madsbox genes. So again, it's a big family, and they have something in common that's known as the Madsbox, which is a special conserved region within all those genes that are in the family. And the sequence of that Madsbox is totally different than the sequence of a homeobox. So we'll come back to that, but for now let's go into this model called ABC model. And this is something that was proposed as a way to explain why do you get the homeotic transformations that you do when you mutate some of these flower genes, these floral meristem identity, floral organ identity genes. And so here's how it works. If we look at the wild type flower structure, from the outside edge to the middle of the flower, you typically are going to see these kinds of structures. So sepals on the very outside. So you can think of this in a linear fashion here, or you can think of it as a, half, like a radius of the flower. Sepals on the outside, then petals, then stamens, and then carpels. And we have genes that are expressed in this meristem, and only in certain regions of the meristem. And we can classify these genes into 3 different groups called the A genes, the B genes, and the C genes. And so within those groups are going to be actually individual genes that belong to that group, but just to kind of lump them together into 3 different categories, we can call them A, B, or C based on where they're expressed. The A genes are expressed in the territories that become sepal and petal. the B genes are expressed in petal and stamen territories, and the C genes in the stamen and carpel territories. So then if you look at either the individual genes or the combinations of genes, you actually have now divided this up into 4 different territories, right? So A alone, that territory usually becomes the sepals. The territory expressing B and A together, that will become the petals. Then we have a different territory for stamens and a different territory for carpels. So by as little as 3 classes of genes only, we can now pattern these 4 different classic flower structures. So what's nice about this is that you can very easily have mutations in any of these genes, and you can make some predictions about what should happen to the flower pattern if these genes were mutant. So let's consider what would happen if we have a class A mutation, and one of the genes in class A is A-petalin-2. And so we're looking at here a flower that has had both copies of A-petalin-2 removed. So these flowers are diploid, so we're just having a homozygous mutant here for A-petalin-2. And what happens in this case is if the A expression is gone, the C expression seems to expand now and take over this whole territory. So this gives us then really only 2 combinations of gene expression patterns. You could have C by itself, or you could have B and C together. And if we just go by our model and read out what that should make, this should make basically carpels, stamens, and carpels again, right? And it's just reading out according to the same old way that it always does. It's just that what's happened here in this mutant background is the expression patterns have changed because the A's have gone away and the C's have expanded to fill the territory. So this should give you a flower structure that resembles carpels on the outside, 2 layers of stamens, and carpels in the middle. And that's exactly what we see for this A-petalin-2 mutation. So this is a very nice kind of validation of this model, and it held up at least for this gene being mutant. And again, this would be considered homeotic transformation because we're converting the sepals into carpels and the petals into stamens. Okay, we could have a class B mutant, and here are a couple of different genes that are in the class B. And by the way, I'm not going to ask you to memorize which genes are in which class, but I might say to you, okay, we have a class B mutant, meaning that the B genes are now not expressed. Okay. So if we have the B genes missing, now we have, again, only 2 different types of territories that the flower should be producing, and so I think that we're probably going to get a lot of sepals and a lot of carpels out of this. And indeed, this is the case. So you can see sepals here on the outside, a double layer, and then 2 layers of carpels. So here's a little bit of stigma and stigma structure. So this also is homeotic transformation, and it also is entirely consistent with the model of the ABC building blocks here. Okay, and then finally a class C mutant. So again, when the class C genes are gone, the A genes expand and fill this whole territory. We get 2 different types of flower organs predicted: sepals, petals, and sepals again. And this time we have, you can see 2 layers of petals for the Arabidopsis flower, which is quite obvious to see. And then also some sepals. So you can't really see the sepals on the outside layer, but these flat structures in here in the middle are sepals. And it's pretty obvious there's now no carpels available with no stigma-like structures on them at all. So this ABC model has been really nice to try to predict how it is that this patterning is happening in plants. And it's very simple and has held up well under hypothesis testing. So good model to include here. Question?
What would happen if you had like both the A and C knocked out because the B regions don't have any area where they act on their own?
That's good. good question. I'll have a slide on that in about 2 or 3 slides here. So, some of the double mutants. So you could predict some of these double mutant phenotypes here, or even a triple mutant, and see what we get. So if you mutate both the A and the B genes, you're only going to get C gene activity left. The C genes by themselves are carpal, so you should get just a mass of carpals for the entire meristem structure. So even though it's successfully converted from vegetative to inflorescence, when it now tried to take the next step and go on and make a flower, the only thing it could make was just carpals. Not a very beautiful looking flower. B and C being mutant together would leave you only A, and that would give you a flower composed only of sepals. And then, so here's your question where we've mutated A and C and only B is left by itself. And this is sort of something that doesn't really happen in the wild type case. Normally there's no territory where only B should be expressed. B is always expressed in combination with C or A. And the phenotype that you get is somewhat intermediate between petals and stamens. So it's kind of taking a step towards the bee-like characteristics, but not being fully perfect in the way those structures are formed. And then you can think about what you would predict for A, B, and C being mutants all together. Any ideas on what that's going to look like?
No flower.
No flower at all, basically just vegetative structures. So just kind of leafy structures. So even though we had this, territory set up, and we're not able to do anything further with it, and it just became leafy structures. Okay, I tried to find some pictures of these, but they're not really great pictures. But here's one where we have now a double mutant between apelin 2 and apelin 3, and this eliminates B and A activity and leaves you only carpals. So you see this massive carpal structure with this hairy stigmas here and there. Then here's the double mutant for the B and C, leaving only A activity, and that's going to be a mass of sepals. So it just looks like kind of an overlapping leaf-like structures there. Here's one that's these characteristic structures of somewhat like petals and somewhat like sepals, but not really completely convincing either way. And then here's your leafy structure that is the result of all 3 of these different gene classes being mutant. All right, so these Matchbox transcription factors, what are they? So these Matchbox are a set of transcription factors that are active in plant floral meristem, floral organ patterning. And I'll show you a slide here to show you which ones in the genes we've talked about so far are Matchbox genes, but several of them are. But they're not only in plants, they're also found in animals as well. And originally, people started to find this class of genes that they could recognize by having this conserved Matchbox domains, but they had no idea what the genes did. And so they just took the first 4 genes that were found that had this conserved region and took the first letter from each of those genes and came up with MADS. So here's some different genes. MCM1, A-gammas happens to be a plant 1. Deficiens happens to be a plant 1. Serum response factor is an animal 1. And so taking the first letter just spelled MADS, and that became the name of this class of genes. So in addition to the MADS box, they also have another conserved region called the K box. And these things, being transcription factors, do bind to DNA. And in all, there's a lot of these genes in the Arabidopsis, about 80 total. And a few of them, maybe, 10 or so, are involved in the formation of flower structure. Okay, and here I've shown you these ones in red are Matchbox genes. So you can find representatives within the floral meristem identity genes, and then also within these floral organ genes, including the B and the C classes. So they're fairly well sprinkled around in there. So the Madsbach genes, like the homeobox genes, some but not all can mutate to cause homeotic transformation. So some of these Madsbach genes are homeotic. Okay, I think that's our last slide. Any questions on this? Yeah.
With that last slide, was the K-box the domain that's conserved within Madsbach genes? Or that's a separate gene we're talking about right there? Or that's just another domain that's in Madsbach genes?
Okay. So this structure that I'm showing up here in the box with all the different colors, that's the open reading frame for one gene. And N stands for N-terminus. MADS is a sequence of amino acids that's highly conserved in all the family members. I is some sort of intermediate domain, less well conserved. K is another set of amino acids still within the same gene that many of these Madsbach families have, and then C is the C-terminus. So if you bind up all the Madsbach genes, you're going to see the highest similarity in the Madsbach and probably the K-box, but definitely the Madsbach. Any other questions on this? Okay, so right now it's 8:32. Let's come back at 8:42, and I'm going to hand back your exams, and then we'll have a review session.